Hey guys, welcome to chapter 8. This is the chapter where we're going to start to really put together a lot of those things that we've learned in previous classes. We're going to get to see how we actually get DNA sequences and what it means to study genomics. So, what is a genome and what can we do with it? A genome is effectively all of the DNA that an organism has. So, we're thinking about all of our chromosomes, or all of the chromosomes that you can find in any organism. This is just the full set of DNA. So that's basically the definition of a genome. So when we think about doing things in a genome context, we think about doing them large scale. So what can we do with a genome? We can find genes within it. We can search through it for individual genes. We can build resources like databases that other scientists can mine. We can compare one genome to another. So this would be like comparing my genome to the genome of someone else, or comparing the genome of a cat to the genome of a tiger. So you can compare and see what's different. This is particularly helpful when you're studying evolutionary relationships or looking for differences just within even the same species. You can use this to identify organisms. So a specific example of this is that genomic high-throughput sequencing can be used to identify bacteria, the entire population of all the different bacteria in a sample. So this could be a soil sample, or if you hear about the microbiome, this could be even something like gut bacteria that has been sampled from a human. We can design screens that help us tell more about an individual organism. So perhaps a screen for cancer, where we look for genetic differences on a large scale to try and find out what differences are going to be there that could contribute to cancer. So various things like that. This is kind of a 23andMe approach where you're looking for different things. And of course, this leads us up to personalized medicine. We're using what's in a person's genetic code can help us determine a better treatment for maybe a particular disease. And so before we can do any of that with a genome, we have to be able to get the sequence of the genome. So we're going to start out by learning a couple of techniques that we would need to build a genomic resource, starting out small and then working our way up to the more high throughput techniques. So what are some ways that we can sequence? So the first one that we're going to talk about is dideoxy sequencing. And this is like Sanger sequencing, or kind of the standard sequencing that you think about. We'll talk about this one as the basis for some of these other techniques. Next, we'll talk about pyrosequencing. This is not super common anymore, but I think it's really interesting because it kind of gives you an idea of the overall trajectory of this technology from the first time we could sequence something all the way up to these high throughput sorts of techniques that are going to have their own sets of challenges. You can also use this to genotype those single nucleotide polymorphisms like the ones we've been studying in lab. We're not going to talk about this in detail today, but just thought I would mention it. And our last technique is one that is more commonly in use now, and this is called bridge amplification and sequencing. So specialized PCR with a sequencing that follows. So these are some interesting techniques that we're going to learn more about today. So we're going to start out again by learning about our dideoxy sequencing first. So you may also hear this called Sanger sequencing or capillary sequencing. And the hallmark of this sequencing is that we're only going to be able to sequence one molecule of DNA at a time. So maybe not just one molecule, but it could be lots of molecules, but they're all the same sequence. So we're focusing basically on one DNA sequence that we want to sequence. So this could be part of a gene, this could be a cDNA, but we're working on a very small scale. So let's get into some of the interesting features of this. So this is the DNA we want to sequence. You see it's just one set of DNA here. We're not looking at the whole genome. This, of course, is going to be a modified PCR. So this is going to be really similar in principle. There's going to be a lot of things going on with this that are going to be very similar to a PCR with some modifications. So we're going to need some primers. Okay, these are going to be our sequencing primers. They have a lot in common with the primers we use for PCR, but we're going to see that there's some difference in the way that we use them. So the sequencing primer is, again, going to be used to help us target the DNA that we're looking for. You can see down here on the bottom strand, we're looking for something that's complementary, and it's also going to serve as the template to help us synthesize, adding on only to the 3' direction. So again, that's very similar to the types of primers that we would use in a standard PCR. The main difference so far that we're looking at between sequencing, our capillary sequencing, and a PCR is going to be that generally in a PCR, standard PCR, the primers are paired. So you might have a primer like this. We call this the forward primer. We're going to synthesize from left to right. And then you'd have a primer paired with it that would bind somewhere on this top strand. And then we'd synthesize from right to left so that you got both copies of the sequence. In this particular case for sequencing, we're only going to sequence one strand at a time. You can sequence both strands, but you need to separate the reactions into different tubes. That's because once we do this sort of PCR-esque amplification, even though these are complementary, it will make the sequence kind of garbled. So we'll explore that a little bit more later. So effectively, primers are similar. We're doing one strand at a time. Okay. Another difference between our capillary sequencing reaction and a PCR is the type of nucleotides that we're going to use. In a standard PCR, we add our building blocks, our deoxynucleotides or DNTPs. These are our building blocks that are going to have, again, it's going to be a set of those 4 nitrogenous bases, A, C, T, and G. And this, again, just a standard nucleotide we would have in a reaction. In capillary sequencing, we're going to use what we call a di-deoxynucleotide or DDNTP. So if deoxynucleotide referred to the fact that we didn't have a hydroxyl group on carbon 2, di-deoxy means we're going to be missing this second hydroxyl group. We do not have a hydroxyl group on carbon 3, in this case, just a hydrogen. So what does that mean? This is going to give our nucleotides a special property that we call terminating nucleotides. So why does this have this property? So again, in our first PCR example, in a standard PCR where we're not trying to sequence anything, we have the hydroxyl group. That hydroxyl group is actually very important for adding new nucleotides. So this is the growing chain of DNA. So this is our strand synthesizing. This is the 5' start. This will be the 3' end. And we have to catalyze this reaction so that we are going to basically stick this phosphate to this. It's going to end up being positioned off of this 3' carbon. And we're going to ignore the last 2 phosphates. Okay, we'll have a chance to see that up a little bit more closely a little bit later. But the point here is that the hydroxyl group is super important for adding new nucleotides. So if you don't have that hydroxyl group, this is a sad panda if what you wanted was to add new nucleotides. So why does this help us sequence? So I think it's important to know what's in the tube for the sequencing reaction to kind of get the whole picture. So here we have our template DNA. That would, of course, be in the tube, and our sequencing primer that will bind and serve as the template to synthesize from, again, this 3' in left to right direction. In this tube sequencing reaction, a normal PCR would have a set of our regular deoxynucleotides or DNTPs. The sequencing reaction will have these DNTPs, these normal ones, but they'll also have a set of each of the dideoxy or terminating nucleotides. So this means that at any point during the reaction, as we're adding new nucleotides, and note similar concepts here, we're just complementary A to T here, we can either add a normal nucleotide or a terminating nucleotide. If we add a normal nucleotide, we can proceed with the reaction. If we add a terminating nucleotide, this is as long as this product will be. And so we've already learned in lab about a technique that we can use to separate things by size. So that will be the basis of this experiment. So in order to really think about why this size difference actually helps us sequence, we can kind of think about the reaction proceeding through this entire DNA chain. Okay, so remembering that we have a population of normal nucleotides, and then we have 4 separate tubes, and we're going to put in each of those 4 tubes one of the terminating nucleotides. So in this tube, we would have all 4 regular standard nucleotides plus terminating A. In the next tube, we would have all 4 nucleotides plus terminating T. Similarly, all 4 plus terminating C and then terminating G. Again, all 4 nucleotides. So that would mean in each of these tubes, so for our A tube, as we go through the reaction, if we're adding a T, a G, or a C, those will always be normal. If we're adding an A, we have a chance to add either a normal nucleotide or a terminating nucleotide. In the T tube, all of the other nucleotides can be added normally. When we get to a T, we can either add the terminating or normal T. Okay, so all of these tubes are going to work the same way. Why do we care? Okay, so we are going to work this reaction out. Here's our first addition to our primer. This was a terminating T. That means this, I guess, in principle, is only 4 nucleotides long. In reality, it's a little longer than that, but you can see that it's really short. Run that on a gel, it's going to go super fast. So it's going to be at the bottom of the gel. Okay, so this is in the T lane because it's got terminating T in it. All right, so we can check out our next reaction. Here is, we've added a normal T, but a terminating A. And since we know that the terminating A's are hanging out in the A tube, we're going to run on this gel. We know it's going to be in that lane, but it's going to be slightly larger than the one from the previous T tube. Okay, we can do the same thing with C. Note it's slightly longer. This is an interesting one because we've had already kind of some time to spend in our T tube up here. This is where that chance is really illustrated. So for this first pairing of T to A, we could have added a terminating T. We'd be back at this size. But because we do have a population of normal nucleotides there, we can add this T normally and proceed. And each time we have a T, we have a chance to add a terminating one. So we're going to be in that same T column, but again, each one's going to be slightly larger. Okay, we can then add a G and then another G. Okay, so move this down just a little bit to give us some more room. We can add this G, and then our second one will be another G. And so you can look through all of these, take some time, pause the video, and check it out. This guy's a little shifted, but that doesn't have a lot of meaning. That was just me being lazy at PowerPoint. So you can see how we've added each of the things. We've got a T, and column T again, A, and A. And again, you can see how each time we added these T's, this particular product, we were able to add a normal T at both of these first two, but then terminating at the third position. So this will just be longer, and you can see each one is slightly longer than the next. I think it's important to point out here that most gels do not do a one nucleotide resolution. These have to be run on specialized gels that allow for that level of resolution. So we can really see this difference, okay? And when you read these sequencing gels, you can start at the bottom, look which row it's in. So we know T, A, okay, and we're starting from 5' end. T, A, C, T, G, G, T, A, T, C, A, A. And you can see how that kind of plinkoed out, okay? So this is how people originally sort of did this based off of size. It still works this way, but we've streamlined it a little by adding fluorescence to each of the nucleotides so that they can all be done in one tube, and the resulting gel would look something more like this, where you'd have these same products, but since these are labeled, the terminating nucleotides are labeled specifically, you're able to do the reaction in one tube, one lane, and then the computer can read these fluorescence patterns as they run through the gel. So what do you end up getting? What does this data look like? So this would be an example of data you would get from a sequencing run. And these peaks correspond to the computer reading the fluorescence of each of these. So green, red, this one's black, green, blue, green, red, and each one of these corresponds to a nucleotide, and the computer will put these together for you. So you can see kind of how the results work. So important things to note about capillary sequencing, of course, will be things like making sure to remember that this is for one kind of homogeneous population of molecules. Because you need primers, you're going to have to know a little bit about the sequence to begin with. And we're going to be using terminating nucleotides. Otherwise, this is fairly similar to just a general PCR. We just need an instrument to help us read the fluorescence afterwards. So now let's talk about pyrosequencing. So capillary sequencing, which we've just covered, or dideoxy/Sanger sequencing, is still commonly used. It is part of the repertoire of tools that we can use to sequence a genome. We're not going to use that type of sequencing to sequence a whole genome from scratch anymore. We're going to be doing some other things, but we do still use it. I think that's important to note. Pyrosequencing, while there may be some people that do use this a bit, it's not as in vogue as it once was, but it is one of the techniques that was sort of one of the more early methods of getting more high throughput sequencing. And it's going to allow us to get that sequence without having a lot of prior knowledge of the sequences. And that's one of the interesting things. Okay, so first of all, I think it's not shown on this slide, but it's important to understand that at this point, we're doing high throughput sequencing. We're getting ready to sequence on the genomic level. We're trying to get as much of the genome as possible sequenced here. So this means that you could take all of the chromosomes in whatever organism you're studying, and you can break the DNA up into small pieces. These small pieces can then be individually sequenced, and we'll see how that works on the next couple of slides. For now, let's learn a little bit more about this reaction that we're going to be doing. This is a really interesting technique. This just really works in a way that I don't know if my brain could have come up with this. This is kind of like the opposite of what you'd want to pay attention to when you're going to be doing this type of thing. But we can add 4 enzymes, DNA polymerase, that's standard. We had DNA polymerase in our prior reaction as well. I just didn't talk about it because it's pretty much, if you're doing a PCR-esque task, if you're synthesizing DNA, there's a polymerase there. ATP sulfurylase. Okay, this is going to be an interesting enzyme. We're going to see how that works. That's going to help us. kind of convert our products in this reaction. And luciferase, you may have heard a bit about this. This is going to be an enzyme that actually helps us produce light from ATP. So that's what we're going to be looking at. This is the same enzyme that's present in fireflies, make them glow. And then apyrase we're going to be using to kind of clean up some of our reaction space. So remembering now how our DNA synthesis works, we've just seen this on a previous Sanger sequencing slide. But as the DNTP is added, last time we were really focused on this hydroxyl group. Now I'm a little more focused on the phosphates, which hopefully you've gleaned that from the fact that I've made them inappropriately large. Normally this phosphate would join. Okay, and of course I've been lazy with this, so there's going to be actually some bond rearrangement, and this is actually going to be a covalent bond. But what we care about right now is actually these two released phosphates. Okay, so this is called pyrophosphate. And again, this happens anytime we add nucleotides. Okay, this is just going to happen as we synthesize DNA. It happened in the last reaction. It happens during natural replication. It just does. But most of the time, we're not really interested in it because we're interested in this growing strand. Okay, so here we've got these two pyrophosphates that we normally ignore. They're going to be utilized now. What we're basically going to do is use those as a marker. Kind of ask yourself a question: did I add nucleotides in this reaction to the growing chain? So did I connect this and add a new nucleotide? If the answer is yes, pyrophosphate is released. If the answer is no, you will not have pyrophosphate. So you'll see that the whole thing hinges on that. Okay, so anything that we incorporate will give us our pyrophosphate, but anything we don't incorporate will still be these nucleotide triphosphates, DNTPs, and these are just regular DNTPs. In this reaction, we add them one at a time. So first we add, a pipette full of C, then T, then G, then A. And between each of those additions, we have to get rid of the extra. So now we're going to make this question a little bit more specific. I added, a pipette full of A, or adenine. Did I add it? If I did, we'll get the pyrophosphate and we'll add that base to the chain, get rid of what's left, okay? And then we'll add the next nucleotide. So we're going to see this in a little more detail, but that's what apyrase does. It's actually going to clean up this reaction and get rid of extra nucleotides that aren't incorporated into the strand. Then our ATP sulfurylase is going to convert pyrophosphate into ATP. Okay, so these two phosphates, we're now going to reconnect this to make an ATP. Okay, so this is why we care about the pyrophosphate, remembering that if we didn't add the nucleotide, then we will not get pyrophosphate and we will not make ATP. ATP is going to be, again, used by luciferase to generate light. So basically, we're asking, I added a bunch of A's. Did I incorporate them into the growing strand? If yes, you produce pyrophosphate, which is turned into ATP, which is used to make light. So you get light if the answer is yes. If no, we degrade them with our apyrase and you get no light. So this is a really interesting technique. What happens is we can capture kind of an image of what's happening between each of these additions of nucleotides. We add a pool full of guanine. If it gets added, we see flashes of light. If it doesn't get added, then we're not going to see any light. And we kind of have a computer that will take a picture of that before we wash all of the extra away. So basically what we're looking for is light. Okay, so why is this high throughput? We've got kind of the mechanics of this down, but how does this become high throughput? Okay, so let's take a look at this again. So at the top, there's a lot of things up here going on that we've just described on the previous slide. So let's start there and then work our way backwards to the left. So again, we had our deoxynucleotide, and these are normal, they're standard nucleotides. We add them. Whatever we need is incorporated into the strand. It's incorporated here because we've paired it with this G that we're trying to sequence, okay, with DNA polymerase. From that reaction, we've produced pyrophosphate. We destroy the extra CTP, okay? Then we're going to use that pyrophosphate to make ATP, which will make light. So did we add this? Yes, light. Okay, we wash away the extra DCTP, and then we add the next nucleotide. If what we add is G, it will pair with the C, and we'll complete this process again. If we add something else like A, it can't pair with C. All of those nucleotides will be degraded. We won't get any pyrophosphate. We won't get any light. Okay, so we can think about whether we got light or not. And then when we have these two C's in a row, if we add guanine, we're actually going to see that we get double the amount of light. So even the intensity can be measured. So I think it's important here to look at the actual data. All right, so this is the way that data would come from one of these reactions. Okay, so at the top, we're going to have the actual sequence. At the bottom, we have the nucleotide that we added. So notice it's kind of like a repeating pattern. We add A, then we add G, then we add C, then we add T. Then we start over A, G, C, T again. So first we add A. Nothing happened. There was no light. That meant we didn't add it. So that wasn't what we needed to add. All of those get degraded. We add our G. G did get incorporated. It paired with C, like something up here. Okay. So actually we can start at the beginning. So our G was incorporated. We had a spike of light. Next we added C. That was incorporated. You can see that it paired. A spike of light. Next we added T. T is not the next nucleotide that we needed. We needed A, so nothing happens. When we do come back around and add A again, it gets incorporated and we get a peak of light. Notice we've got double. We've got two G's we need in a row. We add two G's, we get twice the pyrophosphate, which means twice the ATP, which means twice the light. Similarly, we add these C's and we get a dual kind of double height peak of these C's. Okay, we can return T and then of course AG and then C. This is that C that's purple up here. Okay, and then again back to this double G, and you could go through this whole sequence. I think this makes a lot of sense once you sort of know what's going on. And there's a lot of complex chemistry happening, but at the same time, if C was the next nucleotide, you get light. If it wasn't, you don't. So you can really boil this down to a more simple thing. And again, we still haven't answered that question. Why is this high throughput? This is sequencing one molecule just like our previous thing. The technology behind this technique means a couple of things. First of all, let's look at this bead and primer situation. So if you remember, I told you that all of the DNA for this experiment would come from the full genome and it would be broken up into small pieces. So the small piece is this blue fragment. Okay, the blue fragment where you can actually see the sequence start. Okay, the blue part of this where you don't see sequence, this would be kind of like an adapter that we already knew the sequence of that's attached to a bead. Okay, what's the bead for? We'll talk about that in a second. We have control and know what this blue sequence is, so we can make primers that attach to it, and then we can ligate using similar ligase from DNA replication. We can actually just glue the mystery sequence that we're about to find out more about onto this kind of adapter area. And then the primers we add, we only have to know what this adapter area that we added would be. We don't need to know the actual mystery sequence. So that allows us to sequence things even without any prior knowledge of the sequence. So that's one way that we're going to start adapting this to be more high throughput. The second thing is that we're going to break the genome up into lots and lots and lots and lots and lots of pieces. Each piece will get stuck to its own adapter bead. So you could imagine like jillions of these with different sequences attached to them floating around. Okay. Each one of these beads kind of gets put into its own tiny well. So think about those 8-well strips like we use from lab, but much tinier. Each one of those beads goes into its own individual well. So the reaction is performed on each of these molecules separately, but at the same time. Again, a computer takes the pictures and it says, oh, here's light, or I didn't see light. And then it kind of analyzes the data and spits the sequence out at the end. So this is a really cool technique. Again, I don't know how many people really use it now, but I think it's interesting to talk about because you can see this is one of our first techniques that's called sequencing by synthesis, which basically, in my mind, means you're kind of watching this happen and recording the nucleotide you added at the time that you added it. So to me, that's a pretty cool kind of technology. Okay, so we've got pyrosequencing, we've got our capillary sequencing, and so our next thing that we're going to need to talk about is some more high throughput sequencing techniques, bridge amplification and sequencing. So bridge amplification is not sequencing. It is the first step that we're going to take toward sequencing. So this is going to be our first step toward sequencing. So a lot of things here are going to be a little bit normal. Okay. So let's take a look. And this is a pretty static image, but I've got some more animated stuff coming up next. Okay, so we have here the DNA that we want to sequence. And again, this is the genome and it's broken up into lots of pieces. We're going to have adapters. Hopefully from the last section on pyrosequencing, you kind of get an idea what those are for. Okay, in this case, these are going to help us kind of serve as places for primers because we know what these adapter sequences are. They're also going to help us stick to the specialized amplification area that I'll be telling you about. Okay, so the first thing we do when we get these DNA sequences broken up, and this can be broken up a lot of different ways, can be you can actually use restriction enzymes to break them up. You can actually use sonication to break them up into random pieces. So there's a lot of different ways to go about that. We can then ligate or glue these adapters onto each end. And then these are going to serve to help us adhere to the specialized slide. This specialized slide is called a flow cell, and it looks pretty much like a microscope slide if you don't know the difference. Like if you're not looking under a microscope, it looks pretty much like a microscope slide. So what you're going to see is that we've got a lot of these little areas where our DNA sequences could stick on this flow cell. So notice this is going to be complementary to our purple adapter. Okay, and we can kind of glue these down. All right, so this little piece on the flow cell helps make what we call a bridge. So this is 100% a PCR. So we're going to see that we're going to amplify this. We're going to have a primed reaction. We have to amplify it, but it's done in a bridge formation. So otherwise, this is a very normal PCR. So we've got all the standard nucleotides there that we need, and we just amplify. Okay, so you're going to see that we actually just amplify it. The idea is that we're doing this to generate enough of the same molecule in a place to amplify the signal that we're going to try and get in a minute. So notice we've had a couple, like 4 pieces of DNA up here. We've gotten them ready. Each one of those pieces are different. They're different sequences, and they can stick to different places on this slide. So what we're trying to do is amplify kind of a spot for sequence A, a spot that's full of sequence B, a spot that's full of sequence C, and a spot that's full of sequence D. You can see that illustrated here. Within this set, this is actually technically called a cluster, in this step called cluster generation. Within this cluster, all of these molecules are identical. They're just like a PCR curd. Okay, within this cluster over here, they're all identical. Within each cluster, it's identical, but they're different between clusters, different sequences. So again, the purpose of the bridge amplification step was not to sequence anything. We haven't actually done that yet. It was to amplify the signal that we're about to try to get. Okay, so I think so far it's been a pretty normal process, just done on a slide shaped like a bridge. Okay, so how do we actually get the sequence from this? Okay, this is probably one of the most interesting things that I learned about when I was in graduate school, but the technology here is that we're going to use nucleotides that are special in a couple of ways. First of all, they're all fluorescent, so they're all going to be colored depending on what nucleotide they are. Second of all, they have a property that's referred to as reversibly terminating. So the terminating part of this means that you can't add anything to it. So we can only add, even though we add the whole pool, the whole set of 4 to do the sequencing, you're going to be able to add only the one that you need at that time because it's terminating. But then once you get ready to go to the next step, you've recorded that information down, you reverse that property, you can now add to it again, so your next nucleotide will be added to it. So what happens is, let's take this cluster back here because they've actually magnified it. The first nucleotide that will be incorporated here is a G. Sorry, a T. I'm actually looking here. It's going to be a T. Okay, so the T's are green in this case. We add the T. That's all we can add right now because even though we've got the whole pool in this reaction, the T was what needed to be added and we can't add to it yet. We get the computer to actually take a picture of this fluorescence, and it's taking a picture of all these clusters at once, but we're focusing right now on just this guy up here. We do that. We reverse this terminating property. We add a new set of all 4 nucleotides onto this whole slide. The next one to get added is red. I'm going by the position here. The first one was T. The position on this slide now will be red. Okay. We do the same thing again. Take the picture. Get ready to add new nucleotides. Add the whole pool again. The next one's A. Take the picture. G. Take the picture. T. Take the picture. And we're doing this for all 4 of these different clusters at the same time. So you could get the sequence of all 4 of these. So if you started over here, you could say, okay, G, C, T, G, A. If you started at this cluster, C, T, T, A, G. Okay, and you can get all 4 of them. And if you imagine doing this like for all of these different molecules that we could have possibly gotten from the genome, sequencing them all at once, and then the computer is decoding these images for us. So let's take a look at the animation here. So what we're looking at is, this is going to be the DNA sequence that we want to sequence. Because we've put these adapters on, we don't need to know anything about it in order to prime the reaction. We can use this paired primer on the flow cell that we already know to prime the reaction, and then we can just sequence the part we don't know about. So once we've done the amplification, we've generated a cluster, okay, and this would be within the same cluster, so they're all identical. We've added our pool of 4 nucleotides. The first one that gets added is red, and because these are all identical, each one gets a red, okay? So these, again, we take the picture. We can't add anything else to this now, but once we've taken the picture, we can unblock these, add some more. We zoom back out, okay? Now, here's the red guy we were just looking at in that cluster. Now we can look at green, blue, yellow. Okay, we can look at all these clusters at once. Remembering that if we zoom out even more, okay, kind of remember this position, green, green, blue, blue, yellow, and then red way over here. To the computer, these just look like dots of light. So between each cycle, it takes a picture. So A, T, C, and you can keep track of all of these at one time based off of what colored light is flashing. So these are going to actually usually sequence, I think on average, people sequence maybe about 200 nucleotides at a time. But that means that what we're going to have to do is actually add these back together at the end. So this would be the part where we've taken our cells from whatever organism, gotten the DNA. So this could even be like the cheek cell DNA we extracted in lab, broken into lots of small fragments. Okay, and you can control the size of all of this. But, you know, the kind of in between here can vary. But once we have all these short, this would be kind of like the computer taking those images, the short sequences that we get back from this, we're going to have to put them together. So this is why we're going to be leading into sort of a more computational approach in this class, because this is where genetics really meets computational tools. Once we start dealing with all this data, and like, listen, we're talking about like terabytes worth of data here, like millions of sequences. So you're not going to want to put those back together by hand. So computers can help us do this. And you can just add like a splash or two of our capillary sequencing just to get some longer sequencing to help place these. We're not going to directly be doing this in lab, but we're going to be learning a little bit more about this process. So we've got a lot of different techniques that we can use. Next couple of weeks, we're going to be learning about different types of RNAs and how we can use this technology on RNAs as well, and how we can use it to help us find genes within the genome.